All right, it's my pleasure to introduce Joe Ayers. Joe, let's do the formal part first. He was an undergraduate at the University of California. Well, he did most of his pre-faculty work at UC. This was in Riverside. Then he moved to Santa Cruz to do his PhD, still University of California. And then, if I'm not mistaken, you went to Marseille, France, to work with Francois Clarac first. And then back to San Diego, to University of California in San Diego, this time to do a postdoc with Alan Silverstone, before moving to where you are right now and have been ever since. Am I right?
Indeed.
Okay, so Joe has done a lot of things, but I have a few things that I want to ask him maybe to dispel first. So first, rumor number one, you're a beekeeper or have been.
My best recipe is honey smoked lobster.
Ah, good. And on this side, he has done some honeybee robotics or participated in it, right?
Yep.
So you're a real and a virtual or whatever robotic beekeeper. The other thing that Joe has done is he became a chef and published a book. And I haven't done any of these recipes, but this is not a rumor. This is true. You can buy it in Amazon and other places as well. Another rumor here is that you have been enshrined at the Smithsonian Institute, but this might be actually true for your robots rather than yourself, but you have been exhibited, or your robots have been exhibited there, right?
Yep.
So that's another one of the things he has done. And then I understand also that you were a lobsterman in your youth. Is that right? Or your son is?
No, my father was a commercial fisherman in Los Angeles, and I grew up fishing, but I didn't like getting up at 4 a.m.
So I found this here, which is a book that you wrote on the area of Nahant. And first, under the letters, I thought, oh, this is Joe, but these are not lobsters. But no, it's not you, is it?
No, it's largely about Louis Agassiz, who lived in Nahant, and he was the founder of the Museum of Comparative Zoology, and all of the type specimens of North Atlantic intertidal animals were collected in Nahant.
So what does the C stand for in this book?
Excuse me?
What does the C stand for in this book? It's a pun or something?
Oh, my co-author dreamed that up. That was his idea.
Okay, and then another fact here is that Joe works in a bunker, and he has actually renovated that bunker. And we were just talking about it. It's a humongous lab. I once visited it. I was really impressed by the thing. You still have the rails running along the ceiling through which the bombs were transported to this place where they were shot from, right?
Yep.
So that was an interesting visit. And then fact number two, though, is that Joe did work in STG with Alan Silverstone in San Diego, published a few papers on control of pacemaker phasing and frequency. That very old paper now, but kind of a classic. And then went on to work on crustacean locomotion, first in Marseille with Francois Clarac. And actually, this is before already when you were doing, this is your PhD work, right?
Yes, indeed.
Anyway, so Joe has been dedicated to the ocean in many ways, and locomotion and crustaceans and other creatures that I think we're going to hear about today. So welcome.
Okay.
Stop this and it's all yours.
Okay, sounds good. Okay, that should be starting. Okay, can everybody see that?
Yeah.
Good. Okay,
yes.
so I started graduate school in 1970, and the first book I read was by this guy, who was really one of the founders of what we now call artificial intelligence. And he wrote a book just before he died called Embodiments of Mind, which, if you've never read it, it's just really worth taking a look at. But he had a sidekick named Walter Pitts. And Walter Pitts was sort of the ultimate nerd. And they went together, tried to apply formal logic to the nervous system. And they published several papers on this. And they finally flicked it in and decided that the brain is not a computer. And Walter Pitts went off and drank himself to death. And that was kind of into that. And then McCullough went on to publish papers like Where is Fancy Bread and stuff like that. So that initial start on artificial intelligence kind of went away. But it got revived with Stanley Kubrick's film, which was based on a novel written by Arthur Clarke in 1951. And it was supposed to be about what technology was going to be like 50 years later. Well, that's why it's called 2001. It's now 70 years later. Where's Hal? The idea of sentient computers and stuff like that have just simply not been realized. And when you look at artificial intelligence in general, it's all computer algorithms. The nervous system doesn't have any algorithmic processors in it. It doesn't do things algorithmically. And the way the nervous system works is networks of synapses and neurons. And I call this biological intelligence. And my sort of cognition test is I always ask the cognitive neuroscientists, in terms of neurons and synapses, what is a thought and what is thinking? And you never get any positive answers on what that is. So I don't think this has gone very far. Now, more recently, we've seen a resurgence of network ideas that started with Hopfield and Tank in 1986. And this is figured one, where they argue that the brain is full of backpropagation. But there's no attribute as to where this figure came from, what animal it was, where in the brain it was. They just basically made it up. And what has emerged from that is the computer scientists thinking that this is how the brain works. And they started off with artificial neural networks, and now we're getting into machine learning and deep learning. And, you know, when I first got it started in the robotic world, I started thinking, well, this is kind of the way to go. And I think it's a dead end. And so what I'm going to describe for the rest of this talk is what I call biological intelligence and approaches to biological intelligence that enable us to build a variety of robots. Now, when I was an undergraduate at UC Riverside, I had the great pleasure of having Crellin Pauling, who was Linus Pauling's son, as my academic advisor. And when it came time for me to go to graduate school, I decided I wanted to do neuroscience. And it was unthinkable to Crellin that I would go anywhere but Stanford to graduate school. So he arranged things or got me in line to interview with Don Wilson as a graduate student. And I had the pleasure of spending an afternoon with him. And if you ever knew Don Wilson, he was dressed in a plaid shirt with paisley pants. He was just a really interesting character. And when I told him what I was interested in doing, and I was really interested in understanding the neurophysiology of locomotion, he convinced me that one of his former students, Jack Davis, was in fact doing exactly what I wanted to do. So Wilson had two students at Berkeley, which were Jack Davis and Al Silverstein. So I went to graduate school with Jack, and Jack convinced me that I should work with Al as a postdoc to learn intracellular recording techniques. And he was really dead on that. Now, what Jack turned me on to was the American lobster as a model animal. And the beauty of lobsters is that they have, as we all know, have identifiable neurons. They have very simple circuits. I mean, an insect probably uses 1,000 neurons to do what a lobster does with one. They have very well characterized sensors. They have very simple neuromuscular systems. They have very stereotyped behavior. And in fact, if you blindfold a lobster with some aluminum foil over their eyes, you can pretty much predict everything they're going to do. Almost all the variability you see in their behavior is them visually responding to you. But the real great advantage is you can eat the experiments. And indeed, I ate them all. And my son is now a commercial lobsterman. So I'm not only get free animals, I'm still eating a lot of lobster. Now, the problem that I had eventually got involved in with Jack Davis was to figure out how lobsters use the same neural circuit in the thoracic ganglia to mediate what I call omnidirectional walking. So lobsters can walk in any direction. They can change their posture while they're walking, and they can change their stepping direction on a cycle by cycle basis. So this is really quite a unique CPG. When I got an actual job, people told me if I kept doing what I was doing as a student, that I wouldn't get tenure. So I happened to go to a lecture given by Mel Cohen on spinal cord regeneration in sea lamprey. And that convinced me to switch over to sea lamprey for a while, although the cast of characters that I had to interact with were not the most fun. But sea lamprey have truly re-identifiable neurons. In fact, one of my students, Gary Swain, showed that you can uniquely identify pretty much all of the 1,800 reticulospinal neurons. They're a true vertebrate. They have a thalamus and all those things. The really interesting thing is there's no myelin in their central nervous system, even though hagfish have myelin, lamprey don't. And so their CNS is clear like an invertebrate ganglia, and you can see into the spinal cord and see the neurons to stick them. They have a very simple neuromuscular system. They have very stereotyped behavior. And one of the most remarkable features of them is you can sever their spinal cord and they will recover completely, to the point where after about 4 months, they're pretty much indistinguishable from a normal animal. So we spent almost 10 years studying that. Now, what both of these animal groups bring to the table is a distributed nervous system. They exhibit metamerization, and at least in the lobster, there's a series of mini brains, each of which controls the sensory and motor apparatus of a body segment. Now, working with Don Maynard, Al Silverstone discovered a little ganglion that controls the movements of the stomach. And this sits in the lumen of the ophthalmic artery. So it gets all of its gases by simple diffusion. So as we well know, you cut it out of the animal and it will survive quite well. So this is a Fritz Goro photograph of the stomatogastric, for those who haven't seen this. And the sort of algorithm we used was to pin the nervous system out in a dish of saline, put wires in the individual nerves so we could identify the neurons, and then come in with micropipettes and record intracellularly. So this is a stomatogastric ganglion prepared by my student Lynn Zhu, where she's been able to penetrate 4 cells with intracellular pipettes. So this is really, really an easy ganglion to work on. For those who aren't familiar with this, the way we identify neurons is we penetrate them with a micropipette. We stimulate an action potential, put recording electrodes on the various peripheral nerves, and the branches of different neurons go out different nerves so we can distinguish and identify them from that. When you can record from pairs of identifiable neurons, then you can study identified synapses. And in fact, this is the entire network within the stomatogastric ganglion, is worked out by a variety of colleagues. And the left part of this generates a slow rhythm, the gastric mill rhythm. The right part generates a much faster rhythm, the pyloric rhythm. What was discovered in the 80s was that there is a vast library of compounds which will modify the activity of this network, and it both alters the cellular properties of the individual neurons, but it also modifies the synaptic connectivity, which generates different connectomes. So this is really, in that regard, totally different than a computer. Now, what has come out of about 50 years of studying these kinds of animals is a general architecture for innate behavior. And when I talk about innate behavior, that's the behavior that you're born with. We can all learn how to play a musical instrument or speak a different language, but we all walk in human. And most of these lower animals really only have innate behavior. And the underlying architecture is central pattern generators. And in multi-limbed animals, there's a central pattern generator for each body segment. They're coordinated among themselves by a category of neurons called coordinating neurons, which pass information from a governing CPG to a governed CPG to maintain the interleg gait. And the role of the gait is to provide a pattern of footfall support to support the animal on the pitch and the role playing against gravity. The third category of neurons are command neurons. This is what Jack Davis worked on. And they are the site where the decision to evoke a behavior occurs, and they descend down into the lower nervous system and activate the CPGs to turn them on. Now, the really interesting feature of this architecture is it's conserved among animal groups. So you can take these same components, and the CPG of a lamprey is much simpler than that of the lobster. But if you reorganize them, you can realize a CPG for swimming as opposed to walking. And we were fortunate enough to be able in 2010 to patent the application of this architecture into robots in general. So we kind of own this. Well, I had been working, we had a Human Frontier Science program, and I was working with Al and George Heinzel to try to understand which of these patterns evoked by neuromodulation were real. So I got together with one of our trustees who owned a sonar company, and we developed a sonar telemetry system that would enable us to record from the nervous system in freely behaving lobsters in the ocean. And then we would take the EMG, process data, convert it to a sonar code, send it to the surface, and then at the surface have a sonar buoy that would enable us to listen to their nervous system over the radio while they were running around out in the ocean. So I had a web page describing this project, and I got a phone call from a guy named Alan Rudolph, who was just starting a new program that he called Controlled Biological Systems at DARPA. And he wanted me to take my sonar telemetry system and use it to take control of a giant lobster for remote sensing purposes. And the idea was that we would send sonar commands to the lobster to tell it what to do. Well, having worked with lobsters at that point for about 25 years, I knew very well that under the most carefully controlled experiments, they will do what they pretty much please. And I was able to convince him that it would be easier to build a robotic lobster. And he liked my explanation, and he gave me a lot of money. really a lot of money. And being from DARPA, I learned that they truly operate by the philosophy that if you give 9 women $1 million each, they should be able to give birth in a month. And I think he expected me to do this. Basically, that was the kind of the speed they were looking at. But anyway, we were able to build a robotic lobster, a robotic lamprey through NSF funding. I was also funded to work on the RoboBee project. And we also worked on a biohybrid robot called Cyberplasm. So I'm going to spend the rest of this lecture sort of talking about how we got the robot lobster and the lamprey and at least the helicopter proxy of RoboBee to work. Now, the initial robots that we built were built with what were called finite state machine controllers. And they're a little bit brittle. And it became very clear that if we use that kind of control model, they were going to get stuck. Well, at this point, I was talking to Al about what I was planning. And he had been working with a fellow named Henry R. Babineau, who is one of the Jasons. And Henry was interested in studying the nervous system as a model of nonlinear dynamics. So he asked Al, how many degrees of freedom does a lobster neuron have to enable dynamical modeling? So Al took some, these are the LP neuron from 4 different stomatogastric ganglia, in which he went in with his Lucifer yellow cell cult technique and killed all the presynaptic neurons to LP. And sure enough, when you isolate LP from all sources of synaptic input, it goes into truly chaotic activity. Now, one of my thoughts is that when animals get stuck, they tend to wiggle and squirm. And I'm convinced that what they're doing is they're increasing the level of chaos in the networks that would otherwise generate locomotion. And the chaotic variations on the locomotion are the wiggling and struggling that they do to get out of tight spots. So what the source of the chaos was really, really interesting. So they got together with a fellow named Falke, and Falke had them do some experiments where they messed around with the the calcium pump, inosine triphosphate. And what they found was that when inosine triphosphate was low, they got regular bursting in these neurons. But under high levels of inosine triphosphate, they saw chaotic activity. So Henry dreamed up a form of analysis called local false nearest neighbor analysis, and to determine that in the regular bursting, they had only 3 degrees of dynamical freedom, and in the chaotic bursting, they had 4 degrees of dynamical freedom. So that was kind of an interesting finding. And those 4 degrees of dynamical freedom are the membrane potential, the fast dynamics of the action potentials that occur on the axon, the slow dynamics of the transmembrane calcium currents, which give rise to these slow waves. And then finally, there is a rhythm of the inosine triphosphate pump that generates a 4th degree of freedom. So being physicists, they worked on a model developed by Hindmarsh and Rose and added a 4th degree of freedom and created a set of equations that define those 4 degrees of freedom. Now, Henry imported Misha Rabinovich and his crew, and Misha was the chairman of the Soviet Academy of Sciences and was the Russian expert on nonlinear dynamics. And Russians are really good at building analog computers. And one of their people, Sasha Volkovsky, was a master at building analog computers. So he built an analog computer that solved these equations in real time. And these are the so-called UCSD electronic neurons. And if you plug these into an oscilloscope, because they really capture the dynamics of the lobster neurons, they're pretty much indistinguishable from a living neuron. So these are some examples of one of these electronic neurons configured in a state where it's a pacemaker. And then we alter a parameter here called E, and that turns it into a chaotic prototype. So the way you play with these things is there's a series of trim pots here, each of which is associated with either an internal resistance or voltage. And you use that to tune the neurons to be different types. So these were neurons that had variable chaos. And I spent a sabbatical at UC San Diego, basically building up the circuit that I worked out as a graduate student that enables lobsters to walk in different directions. So this little thing on the lower right is that circuit with the 8 electronic neurons that they had at the time. So what we're trying to control is a central pattern generator, and the central pattern generator mediates this omnidirectional walking. So we did some kinematic analysis of lobsters walking on a treadmill. And what you see here is the difference in the limb kinematics around the 3 major joints that participate in walking for the coxobasal joint, the thoracocoxal joint, and the merocarpopodite joint. What you see here is that cyclic movements of the coxobasal joint underlie the swing and the stance phase of walking in both directions. During forwards walking, the movements of the thoracocoxal joint become coordinated with the coxobasal joint to cause the early, the late swing, and the stance phase. When they walk backwards, those are reversed. So the coordination between the thoracocoxal joint and the coxobasal joint reverses. Now, the merocarpopodite joint during forwards and backwards walking doesn't do much, but when they walk sideways, all the propulsive forces are generated by this joint, and the thoracocoxal joint doesn't move very much. So we were able to identify 4 synergies here, what we call, so this is the early swing, the late swing, and the stance. This is the elevator synergy. This is the depressor synergy. This is what we call a swing synergy, and this is a stance synergy. Now, the way the CPG works, well, I'll get to that later on. So the underlying network, we sort of worked out hypothetically on the basis of all this causality. And the basic idea is that there is a recruiter for each of the muscles that move the 3 pairs of joints. So there's 2 antagonists for the thoracocoxal joint, 2 for the coxobasal joint, 2 for the extensor and flexor. And the elevator and depressor interneurons that form part of the neuronal oscillator directly activate their recruiters. But there are 2 other interneurons, which we call a swing phase interneuron and the stance phase interneuron. And they underlie the swing and stance phase. Now, within this CPG, there is a pattern of synaptic connectivity where the elevator, and this is very much like the pyloric system, the elevator inhibits the depressor and the stance, but it's a stronger inhibition on stance. So there's a delay between when the depressor starts and when the stance phase interneuron starts. And that's what forms the late swing phase of the step cycle. So neuromodulation turns on the CPG, gets it oscillating. And then there are these command neurons which select the direction of walking. So for example, the forward command turns on the synergy between stance and protraction and swing and retraction. And then there's a third connection we call a recruiting connection, and that activates the muscle that produces translational propulsive force, which during forwards walking is the retractor that enables the animal to walk at different speeds. Now, these connections are orthogonal, and there's one for each direction of walking, and that's what causes the shift in the pattern of coordination that allows them to walk in different directions. There also is a connection, a postural connection to the anti-gravity synergy, which enables them to produce a gradient of depression going from front to rear to alter the pitch, or from left to right to alter rows. So this is the total connectivity you need to walk in all different directions at different speeds and at different postures. So I wired these up together, and sure enough, when we turn on the forward command to get all the strengths right, we get a forward walking pattern. If we turn that off and turn on the backward command, we get a backward walking pattern. And you can see the phase shift. These are synergistic during this pattern, and they're antagonistic in this pattern. Now, these control signals are ideal to control muscle, but they're terrible for controlling motors and solenoids and the sort of actuators that most people use to control robots. So I was pondering how to get an artificial muscle. And my brother-in-law found an ad in a magazine called Midnight Engineering for a technology called muscle wire. And muscle wire turns out to be what they make heart stents out of. And the nitinol, which is muscle wire, which is a 50-50 alloy of nickel and titanium, exhibits a phenomenon called the shape memory effect, which means that if you heat it up to a very high temperature, it goes into a remembered state, which is called the austenite state. And then if you cool it, you can deform it, which means if it's a stent, you can compress it down into a tube so you can insert it into an artery. But if it's just a wire, you can stretch it out from deformable to deformed martensite, which is associated with about a 5% length change. Now, when you apply heat, which you could do by inserting these into the body as a stent, and when they're inserting them, they squirt cold saline through the tube that the wire is in. And then when it gets into the artery, the heat of the body causes it to go this transformation from deformed to austenite. But in terms of you do this with a muscle, it will shorten during this conversion from martensite to austenite by about 5%. Now, the way we mediate excitation contraction coupling, and I cannot tell you how important this is, because in the world of artificial muscles, this is the only one that I know of that has a neuronal controllable excitation contraction coupling mechanism. So what we do is we take the action potentials of the electronic neurons, and with a single op amp, we form a comparator that produces a pulse that we use to gate on a power transistor that applies current through the nitinol. So it's during the overshoot of the action potential that current is applied to the nitinol, and that heats it up enough to undergo this conversion from deformed martensite into austenite. And the total parts count there is 2 chips per muscle, and they cost about $1. So this is really a simple mechanism for achieving this. And if you take a weight, and what you're looking at here, this upper trace is the physical displacement of a 1 kilogram mass, and it's being lifted by a 250 micron nitinol wire. And if we vary the action potential frequency, so this is pulse frequency modulation, we can vary both the amplitude of the contraction as well as the contraction velocity. So this is a perfect biomimetic way of achieving excitation contraction coupling. And if we take 4 of these muscles and wire them up to one of my robot legs, we can get it to walk forwards and backwards. In this image, the leg is in a tank of water, which is in front of an oscilloscope. And this is the activity going to the protractor, depressor, the elevator, depressor, protractor, and retractor. And you can see there's patterns changing. And there's an intermediate pattern, which would be the pattern for lateral walking if we were controlling the metacarpal joint. Now, these electronic neurons were incredibly cool, and I really had a lot of fun with them, but they're physically big boards. And to get a bunch of these into a robot is very, very difficult. I spent about 3 years with an engineer, Young Bin Kim, trying to reduce these to sub-threshold analog VLSI. And we could never get it to work. And when we did get it to work, changing the parameters was really, really difficult. So there was another postdoc from Russia, Nikolai Rulkov, who developed an alternative model, which he called the discrete time map-based neuron, in which he has 2 control parameters, X, which is the membrane voltage, and Y, which is a slow parameter. And this is a graph of X in cycle N versus X in cycle N plus 1. And this defines a map, which is the membrane voltage trajectory. So this normally is circulating around this loop here, generating fast action potentials. Periodically, the slow component drags down to this null incline here, and you get a silent period. So over different ranges of X, he had some fuzzy logic. And what's really important here is that these are not differential equations. These are what are called difference equations. And he could get about 2,000 neurons and synapses to compute in real time on a Texas Instruments digital signal processor. Now, this model has 2 control parameters called alpha and sigma. And in the space of sigma and alpha, in this region here, the map defines silence. In this region over here, it defines bursts of spikes. So each of these points here is an XY pair, a sigma alpha pair. And this is the dynamics of the waveform that you see when you configure the neuron to have that sigma alpha configuration. So you can see in this region, you fire bursts of spikes. The duty cycle is varied as you vary sigma. In this area, you fire spikes. And as you vary sigma in this area, you vary the frequency of the action potentials. So this is a really interesting model. And then there's a third control parameter, which is beta, which is the injected synaptic current. And this uses the standard Allen Destex synaptic current model to define that. We also have tunable synaptic parameters. So we can configure this by adjusting the reversal potential of the synapses to be excitatory or inhibitory. And we can vary the synaptic strength and the synaptic time constant to produce a variety of synapse types. Now, when we prototype these models, we do it in a simulation environment called LabVIEW. And LabVIEW is uses graphical programming. And what we do is we connect these different parameters up to a slide pot associated with each parameter and just physically tune the neurons to get the waveform we want. Well, because it's in LabVIEW, that turns out to be the control architecture of Lego Mindstorms. Inside Lego Mindstorms is a LabVIEW engine. So we were able to put a library in Lego Mindstorms that will enable students to control robots with electronic neurons and synapses. So this looked pretty good on the broader aspects of my NSF proposals, that's for sure. But in real robots, we use a digital signal processor that costs about $29. This is a custom-made board by a fellow named Mark Hunt, which is an associate of Ruhlkoff's. And we define a set of structures in C for each neuron and synapse. So we have two categories of neurons. One are spiking neurons that just generate action potentials. The other are bursting neurons that generate the slow weaves and stuff like that. And then the third are the synapses. And so these define the dynamics of the individual neurons, and these define the topology of the network. So when we run this program, we initialize them, and then they just sit in a loop and just update the value of the voltage of each of the neurons and the synaptic currents passing between the neurons in real time. There are no algorithms in this controller. In other words, there's no point where we say, lift your leg, move it forward, put it down, pull it back. All we're doing is adjusting the parameters of these neurons, and they're working it out as a network among themselves. Okay, so what are we going to control with this? I worked with a applied mathematician, Gail Carpenter, and we did an analysis of the swimming of lamprey, where we literally traced their shape. We broke it down into a series of shapes in a movie. We then, for each frame, analyzed the body for curvature. And we define a flexion as a region of curvature maximum. And it has a locus, which is a percent of body length going from nose to tail, and a curvature, which is 1 over the radius of the best fit circle normalized to the length of the animal. When the animal is swimming, these sort into straight lines, which propagate from nose to tail. And we call those a flexion wave. And because they're linear, we can do regression analysis on them, extrapolate them to the nose of the animal. The time between two flexions to the same side is the period of swimming. And the lamprey always look S-shaped because the propagation time, the time to get from nose to tail, is equal to period. We also have an amplitude measurement, which is basically time free, where we plot the curvature of the flexions to the left and the right as a function of position along the body. And you get an envelope of points, which is characteristic of the particular behavior they're exhibiting. Now, we did a really simple experiment where we took a lamprey and we glued the 25% of body length, a little filament of monofilament fishing line, and led that off to a strain gauge. So this number that you see here is the actual instantaneous force or thrust that the lamprey is generating by swimming. If you make a graph of that versus time, you see that swimming is pulsatile. And in fact, if you compare this with our kinematic analysis, you can see that there's a peak of thrust every time a propagating flexion wave gets to the cloaca, which is about 65% of body length. So a flexion wave is to swimming what a step is to walking. And that allows us pretty much to directly compare walking behavior with swimming behavior. Now, Jim Buchanan, working in Sten Grillner's lab, worked out the central pattern generator for swimming in sea lamprey. And the central pattern generator for each segment is 3 pools of neurons, in which there's about 50 neurons in each pool. So altogether, the bilateral central pattern generator for one segment is about 300 neurons. Now, what Jim observed is these neurons fire action potentials quite rarely. So they're really acting as a population. And so we first did a simulation where we simulated all 300 neurons in each segment for all 110 body segments. And we're able to see sparse patterns like you see in the real lamprey. Now, Nikolai, being a really, really bright guy, took a look at the spiking patterns of all the EINs in one segment and saw that they were regularly bursting, and had the idea, well, what if we just average all 50 of these? Which he did. And sure enough, the waveforms he saw in the average neurons looked just like regular old stomatogastric neurons. So we decided, why not see if we can't just profoundly simplify this? And so we built the same CPG now out of 6 neurons. And these are the 6 neurons. And you can see they generate a really excellent replica of the pattern that you see from all 300. And in fact, if we modulate them by excitatory input, which would normally occur from the mesencephalic locomotor region to the excitatory interneurons, we could reduce, frequency modulate the CPG by passing excitatory current just to these neurons. So this really, really profoundly simplified our modeling effort. And we needed also to deal with intersegmental coordination, which Jim had worked out. And that consists of some connections between excitatory interneurons that project rostrally and others that project caudally. And what we found is if we modeled just 5 body segments, we could capture the dynamics of the animal. If we bias the excitation towards the anterior segments, we got a front to rear propagating wave. If we bias it towards the posterior segments, we got a rear to front propagating wave. So this enabled us to get this thing to maneuver, to back up and take off and do all the normal things. And we established a set of neurons and a set of parameters for those neurons, characteristic of a variety of behaviors. Here's forward swimming, here's a left-hand turn, here's backwards crawling. So we were able to create a whole library of behaviors based on a set of command neurons that descended into the spinal cord to turn this on. Now, for a robot to do something useful, you got to be able to control it. And you got to be able to tell it which way to go. So my student, Anthony, started thinking about compasses and how we could put a sense of direction in the robot. So we put a digital compass in the robot that gave us a sense of direction. And then we would compare that with the direction we wanted to go and calculate a statistic, which was the difference between the actual heading and the desired heading. And we call that heading deviation. And we create a voltage which is maximal when the robot is 180 degrees off course and decreases to where it's 0 when it's directly on course. And we use that to modulate a heading deviation neuron. Then we took a very simple motif, which was lateral inhibition with range fractionation, and subdivided that into neurons which fire when it's completely off course and has to rotate and turn around. Another set which is active when it's off in the yaw plane and just needs to turn to one side. And then a region where they can just basically proceed until they need to make another course correction. So we then took these and started building exteroceptive reflex networks. So for each side of the animal, we have a series of commands for backward swimming, for slow forward swimming, for fast forward swimming, and for amplitude modulation. And then we connect those heading deviation neurons to these command neurons. We have also an accelerometer in the robot that when it collides with something causes a rapid deceleration, and that rapid deceleration turns on the 2 backward commands, so the robot backs up and takes off in another direction. So this circuit here, which is our yaw control circuit, I'm going to demonstrate right now. So we're telling the robot to go in this direction. It hits a wall, it backs up, while it's backing up, it's altering its orientation relative to the heading. It then gradually adjusts its heading to get back on course. So there's an example now with simply analog neural networks with no algorithms at all. We can get this to do collision recovery. Now, if we change the desired heading slightly and alter the orientation of the vehicle, we can get it to do wall following. So all we've done here is to change the desired heading, and the animal is adaptively able to negotiate around walls. So we also have controllers for, this is the second duration lobster robot. This is one that's using a finite state machine controller, and it produces pretty adaptive behavior. This is in real time. It's got motors that control the pitch of the claw and the tail to give it hydrodynamic stability. And then this is the fourth generation lobster robot built up by Dan Bluestein. And this is controlled by the same neurons and basic circuits that are in the lobster robot that has the electronic nervous system. I mean, in the Lamprey robot. So there it was rotating by walking forwards on one side, backwards on the other. Here it switches to lateral walking. So this is really quite maneuverable. And this is using a brain now that basically takes a bunch of exteroceptive reflexes for heading, for optical flow, for bump, and then hydrodynamic flow as monitored by the antennae. And that forms the brain circuit, which controls that last robot I was just showing you. Now, the beauty of this thing is the electronics are almost trivial. They're very, very simple. And we brought a vernacular to the Department of Defense, which they had never heard before, which is CHOOP robots. So they have a very low part count. They're easy to manufacture. They're so cheap, you can basically use them as disposably. You can build them in swarms, and they're very highly adaptable. Now, while this was going on, I also got a grant with Rob Wood over at Harvard to build robotic bees. And we were waiting for the longest time for them to come up with a processor that would run on the bee, that we took a helicopter and used it as a proxy for the bee. And so we added some boards that run the electronic nervous system. That's what this is. That has the motor drivers that take the action potential trains and convert them to the current to drive the helicopter. And most interestingly, we had a Bluetooth board on the helicopter, so we could actually telemeter in real time the activity of the nervous system while this thing was flying. Now, if you know anything about bees, which Jorge pointed out, I've had a bit of experience with, they navigate by means of a guide bee doing a thing called a waggle dance. And what the bee does is that when it goes out and finds a source of flowers, it flies back to the nest and it does this dance where it has basically a figure of 8 trajectory where it walks back and forth. And the orientation of the part of the trajectory where it waggles tells them the orientation of the flower source relative to the sun. So that's heading information. And then the amplitude of the waggle tells them how far away it is. So if they waggle intensely, it's far away. If they waggle just a little bit, it's close. So in order to make the robo bee navigate like a real bee, we had to create an odometer. And the way we do that is we use a optical flow chip developed by Jeff Barrows at Sentai. And that gives us a signal that indicates the flight speed of the bee. We then use that to activate an optical flow sensory neuron. And that optical flow sensory neuron synapses on 2 layers of odometer neurons. And They are just leaky integrators. They integrate and fire. And ultimately, the second layer odometer, when it fires, that's telling you that it got to its target. So this is the simple odometer circuit. This is it integrated into the RoboBee brain with the heading control here, the optical odometry control here. And this is a helicopter now, and we're telling it to fly back and forth between these two circles. So you can see it got hit by a puff of wind then, and it slowed down, but it kept track of the distance because it's using an odometer. So it's turning around after achieving the second one, and it does it again. And then it started doing something funny here, and it turned out its battery went dead. So it's pretty robust right up to the end. But this is now the electrophysiology of that electronic nervous system while it was flying. So you can see the end of the first search vector here, the end of the second search vector here, and this is the activity of all of those neurons. So this to me is really synthetic neuroethology. We're creating models of the nervous system that we think we mediate the behavior we see, and we're able to test those models during behavior. Okay, now the thing I want to kind of end up with is what kind of things can you do with this, and what are some of the approaches you can take with this sort of platform? Well, one thing that's missing in most robots, and if you're interested in pursuing search, olfactory guided rheotaxis, or any kind of stigmergy, for example, social insects like ants have 19 pairs of scent glands, and they leave little stinks all over the environment that say, this is where the food is, this is where the babies are, don't eat here, da, 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 da. In other words, they olfactorily code their environment. So a sense of smell is incredibly important for a variety of behaviors that certainly arthropod model robots would need. Now, the way the olfactory system works is you have olfactory receptor cells, and they synapse on olfactory neurons that project into the brain. So we decided to create a proxy for olfactory receptor cells by engineering yeast. So we take yeast, we add some genes in it that cause it to report to a sensor that we have, and then that sensor will generate a sensor current that activates a discrete time map olfactory neuron. So this is our proxy for an olfactory system. And if you're unfamiliar with it, the way synthetic biology works is you take a bunch of gene parts, and MIT has a repository of biological parts where you can get the genes that you need to do this. You build what you call the device construct. You take the device construct and you infect your chassis with the device construct, and then you can add function to the chassis. So in a yeast cell, this stuff in orange here are constitutive proteins which are already present. And one of these is an enzyme called nitric oxide synthase, which turns on nitric oxide, which is the cue that yeast uses for budding. Now, what we added, and Jason Dirks, who worked in my lab and did this, was really quite a genius, is we added the G protein coupled receptor 226, which is the one that enables dogs and rats to smell explosives. And we added the genes for guanide cyclase, adenyl cyclase, what am I saying? Which produces cyclic AMP. And then we added the genes for a cyclic AMP activated calcium channel, such that when the yeast cell smells explosives, it's going to allow a big inward sodium current. And then we can couple that to these constitutive enzymes where the calcium activates calmodulin. That produces what's called activated calmodulin, and that's the activator for nitric oxide synthase. So this is the pathway that we added the function to the yeast to cause it to get a sense of smell. And then for reasons that are just tragic, Jason Dirks left. But the basic idea is that the sensor is the G protein coupled receptor, the reporter is nitric oxide synthase, and the nitrate is nitric oxide. So we then needed to make an interface that went between the engineered yeast and the discrete time map neurons that were the olfactory receptor cells. So I bought this crazy technology called actually it's a set of technologies that we use to build what's called an E-jet printer. And for neurophysiologists, this is really an ultimate toy. What you do is you take a micropipette, and I have a Brown and Fleming, so we can make all the kinds of pipettes we want. You sputter coat it with silver, and then you put it in a mount over a precision stage. And I have a stage that moves in 1 nanometer increments in X and Y. And then you apply a voltage between the sputter coated electrode and the stage, and that electrostatically causes it to calve off a micro droplet. And the droplets that this thing produces are 250 nanometers in diameter. So they're about half the diameter of the lumen of a micropipette. And for comparison, a inkjet printer produces droplets that are about 100 microns. So this produces incredibly fine detail. So this is this thing actually printing. You can see the impact zone of the drops, and this is printing now with nanoparticles of silver and ethanol. And we can end up building parts like this. So this has 3 electrodes on it. There's a reference electrode, a working electrode, and a counter electrode. And we do what's called ampeometric sensing, where we vary the voltage between the working electrode and the reference electrode. And we measure the resulting current between the working electrode and the counter electrode. So it turns out that the negative deflection is the reduction peak, and that's proportional to the concentration of nitric oxide. So using additive manufacturing with the inkjet printer, we can print a array of both photosensors and nitric oxide sensors on a glass cover slip. We can insert that into a mount and inoculate that with a biofilm of engineered yeast. And there we have a biohybrid nose. Now, what if the robot discovers something? If it smells explosives, you got to have a way for the robot to communicate with you. So we use sonar coding. And the way the sonar coding works is we use object programming techniques where we send messages that have a target, a method, and data. So we use discrete interval coding where we discretize the intervals between 2 pulses by typically about 15 milliseconds. So we have now 8 intervals. And then we send 3 intervals. The first interval tells us the target. The second interval, so that might be vehicle 1. The method might be change your heading, and the data might be change your heading to northeast. And then the robot could send back and say, I am here, I smell explosives, and this is what the concentration are. Now, you also need to know where it is. And the way you do this underwater, because you can't use radio, and let me tell you, this is an extreme pain, is with what's called a long baseline navigational array. And so our way of doing this is to put a little reel carrying antennae on a tracked vehicle with basically a walk lid on it, and have that locomount out to create what's called the long baseline acoustic array that's hardwired to a base station. So we then send the robot up. First of all, this will send up an antenna to the surface, get an absolute GPS fix so it knows where it is, and then pull down to the level where the sonar signals between the vehicles are going on and monitor the sonar signals. So the way this works is what we call supervised reactive autonomy. And the way to think about this is the way you would interact with a dog on a walk. Say you walk down by a lake, the dog is being supervised by you. You throw a stick in the leg, that's a supervised instruction. The dog swims out, and while it's swimming, it's behaving autonomously, and then it swims back to you. And that's the way you really want to control robots. So we send the robot on a search vector, which has a heading and a distance. When it gets to the end of that search vector, it enunciates its position. We do a time of flight measurement to the different electrodes, and we can specify the location of the vehicle within about 10 centimeters. We then send the robot a command, which is a new heading and a distance. It goes on that search vector, and at the end, it would normally enunciate its position. But if it happens to, for example, smell the downstream plume from a minefield, then it would orient into the direction of flow of water and do odor guided reotaxis and home on the mine where it could blow it up, or it could just go on randomly searching. Now, I want to end with kind of a killer app, which is what my last proposal to use these robots was all about. This is a bridge going from Salem, Massachusetts to Beverly, Massachusetts. This is about 8 miles from my house. And if you see here, and this is a photograph from a while back, there was an open vacant lot here, which you would wonder why there would be a vacant lot in the middle of a big yacht field. Well, it turns out that this in the late 30s and early 1940s was a company called Metal Hydrides. And the guy there developed a process by which he could use calcium hydride to extract the metal out of metallic ores. And where this worked best was with uranium. And in this plant, they extracted all the uranium that was in the Nagasaki weapon. So these are now, these hatched areas are maps where it's still hot there. And in fact, the wall here failed, and this was opening right into the bay here in Salem, which is in the middle of a huge lobster fishery. So this is a pretty nightmarish situation. So what we have proposed to do is to set up a long baseline array and put a Geiger counter on Robo Lobster and map this kind of radioactivity out. So this is a way that you could take now a completely biological robot and get it to do really something very useful. So having said that, I think I'll end there and I'll take some questions.
Good, thank you. Anybody has any questions? Ansgar.
I just congratulated Joseph. Well, hi, Joe. This is pretty close to application. So going back to the first models that you did on crayfish walking, I think what becomes in particular important is that you at that time already made sort of the CPG drive, the depressor levator, which is actually lift and depress the leg to be the kernel of the operational circuits. What was the reason to do this at the time?
So we did a correlation coefficient between the period of all the muscles when it walked in different direction and at different speeds, and especially when it was changing speed. And all the other muscles were most highly correlated with changes in the period of the elevator. So that was
Okay.
what led us to that in the beginning. And that, I think in my first paper, that's one of the figures.
Mhm. I think that comes about in what it takes a leg to move in stance and swing in the end at all.
Yeah.
Yeah, thank you.
The
Super.
crazy part is that was 50 years ago.
I know, it's 400 years ago.
Mikey, I think you had a question. Somebody else had their hand.
Not me, sorry.
Mike?
This is kind of a very abstract, almost kind of career question. Is you had this model that was 200 neurons, and you eventually reduced it to something like 7 neurons.
Uh-huh.
And it kind of reminds me of the way I go about analyzing my data a little bit. Is there any way to know the simple method before you go through the actual complex looking back?
No. In fact, we spend a lot of time figuring out, first of all, could we get away with 5 body segments? So we modeled all of this with starting off with, I think, 10 body segments and kept moving down and down until we got the optimal behavior with 5. And the number of neurons, you know, that's just the difference between vertebrates and invertebrates. Is vertebrates tend to use really sparse coding, and invertebrates tend to use high levels of activity in a few neurons. And it was just a matter of figuring out where the trade-off optimized there.
So it's not something you have to do it the hard way before you can do the simplifications.
In retrospect, I didn't find any simple way of doing it.
Okay, thank you.
Yeah.
Joe, I have a question. So, of course, when you talk about these things, depending on the task that you assign to these robots, you have a certain level of precision that you need or that you can get away with. And the one application I was thinking about is to autonomously driven vehicles, like the Tesla and whatever is being tested out there. To what degree could you apply this kind of approach, the non-algorithmic approach, to something that requires a very high level of precision, like there, in that case?
Well, that's why I'm building lobster and lamprey-based robots. I have to tell you, when you start doing flying robots, you realize that the most important thing is to buy the cheapest helicopter you can, because we have a trash barrel full of them that it crashed and burned. And that's the only example of real destructive testing that I ever had to do. And that's a really hard problem. You know, it's a really hard problem. And you know, the guys that think they know how the brain works are my favorites. You know, they if they think they can control this with brain-like mechanisms, they must be able to, because we can do it. You know, and there's just an issue of finding the way to do it. But really discretizing everything and reducing it down to this highest level of simplicity you can get and still have it operate, which for me was the key in all of this.
Yeah.
In fact, when I started graduate school in 1970, the only course they made me take was a course in electronics. And the guy that taught the electronics course came in, and the first thing he wrote on the board was, "Everything is a compromise." And boy, was he right.
Yeah.
So all of these solutions that we achieved were the best possible compromise we could come up with.
So you started your talk with this position that AI is an exclusively algorithmic, or people who do AI use exclusively algorithmic approaches these days, right? as if suggesting, well, first of all, it's not a biological approach, yes, but as if suggesting that this is perhaps not good, but at the same time, we humans or animals are very imprecise, right? We are really fuzzy about the things we do. We commit mistakes and we're not necessarily very accurate. We make decisions fast and on the fly and so on, but we're not very accurate. But algorithmic approaches can be pretty accurate. So maybe AI is better off being the way it is, algorithmic in that way, because it can be much more precise. Or am I completely wrong?
Well, no, for the time being, until we get a better sense of how the nervous system does these estimations, you're probably right. I mean, we still in these robots have a controller layer that's in between the commands, the sonar that we send to the robot, and actually turning on the command neurons. So, you know, we still have a digital level in there, but that's largely because we're doing acoustic communications. And you know, that's what we got for data is as an acoustic signal. So, you know, I think the more we learn, and I think we'll learn more by doing these sorts of embodied simulations. I mean, this all started off for me writing a simulator to simulate the stomatogastric motor programs so we could test the bandwidth of our sonar telemetry system. Then everything else, once we got that simulator working and then wired it up to some actual muscle, it was mind-boggling how well it captured the actual behavior of the animal. And when you think about changing walking directions, changing height, changing posture, all at the same time, that model does a really, really good job.